everybody. My name is Dr. Marc Schuckit. I'm the Director of the Loyola Institute for Brain Research, and I'm very excited to have a phenomenal WKW Foundation speaker today, Dr. Marc Schuckit, who is visiting us from the University of California, San Diego. There are very few people that I could say are probably at the very, very top of their field and are so knowledgeable that it's almost like a walking encyclopedia. Dr. Schuckit is probably the most accomplished alcohol researcher in the country, probably in the world. He has done amazing studies, longitudinal studies with individuals with alcohol and alcohol use disorder, and he'll tell you much more about this on He has a very distinguished career. He received his medical degree at the Washington University in St. Louis. But really, over the last 40 years, he has made his home in California and has built a tremendous group there. He's worked in the San Diego VA. He was heading the alcohol and drug treatment program there. He's treated a lot of forward-looking treatment approaches. And over the last few years, he's extended some of this to other populations, which you'll hear about. As you can imagine, he's been on numerous committees. He was very pivotal. In fact, I think he's probably the single most important person in how we actually talk about the use disorder disease, because he has done a lot of studies looking at how do we classify, how do we categorize people with various substance use disorders, and what are the criteria that we're using. Early on, he was involved in a multi-site study that looked at the genetics of alcoholism. He identified genetic factors that are risk factors for alcoholism, and he's published several hundred peer-reviewed journals, peer-reviewed articles in very highly journals. In fact, he wrote most recently the most definitive review in the New England Journal of Medicine on the opioid use disorder. So I think you can gauge that this will be a very, very impressive and very insightful lecture. So I'm very happy to have Marc Schuckit here, and I want to warm welcome.
It's lovely to be here. I've had such a good time these last 2 days meeting with people from LIBOR, and it's a real treat. You have a program here to be very proud of. Before I begin, though, I'd like to ask a few questions about the audience so that I know whether I am meeting the needs of the majority of people here. The first question is, how many of you do research? So it's a large proportion of the people who are here. How many of you are, and for those of you who do research, don't raise your hand again, because I want to know how many are not doing research but are primarily involved in clinical care. And those are probably our two largest groups here. That's fine. And then I imagine the other people here are coming from a variety of backgrounds with either some level of interest in research overall or some level of interest in alcohol and drugs. Well, today I'm going to talk about a series of developments in my own career relating to research. And for reasons of happenstance, my career ended up focusing on alcohol and drug use disorders, but especially alcohol use disorders. And in the slides you see, alcohol use disorders will be abbreviated as AUD. And just so that you know where I'm coming from, this is San Diego, and not far from the university, not too far from our home. And that I'm using this because this happens to be the year of our celebration in our department of psychiatry of our 50th year. The first thing that I'd like to tell you, which is important, is to the best of my knowledge, I have no conflicts. Okay. I happen to like Chardonnays and Cabernets, but I don't think that disqualifies me, and I don't receive any money from pharmaceutical companies. I'm not saying that's bad, I'm just saying I don't. And so, as far as I know, I have no conflicts. So, I'm going to talk about what became the San Diego Prospective study. And as I was meeting with people around here this morning, one of the wonderful questions I got was, your career looks so linear, like you knew what you were doing and that's the way you, no, contrary. No, what happens is you get interests, things pan out, things don't pan out, you get criticized for X or Y, and then you, in a paper you publish or whatever, and you kind of veer a little bit that way and then a little bit that way. And if you had asked me in about 1970, when the basic part of this work began, I would have had no idea that I was going to be here talking about the things that I'm talking about today. It's just how things develop. And for this particular study, in 1978, it was when I was at the University of California, San Diego, we picked average of 21 years old, 18 to 25, students. There were 453 of them. Half of them were family history positive, abbreviated as FHP. They had an alcoholic father. I chose not alcoholic mothers because I wanted to avoid fetal alcohol syndrome. So these were students, 453. About half of them had no family history, half of them had family history. My subjects were initially males, and they were evaluated for a bunch of different things that we hypothesized would be different between people with family histories and those without. Then, and you'll learn what those things are. And then once we had them, we had some interesting findings. So we figured, well, we've got what they were like at an average age of 21. Let's keep following them up. And because I am fairly obsessive, we followed them roughly every 5 years, and they're now age 55. And I know their trajectory. I know who developed alcohol use disorders, who developed drug use disorders, who became psychiatrically ill, and have all the data of what they were like when they were roughly about age 21. And as we were following these people up, as they married, their wives entered the study. About 90% of the wives agreed to participate in the study. And as they had children, we gathered information on their kids from the time that they were born and started to interview those kids at age 12, but especially became interested in those kids once they were hitting about age 18, because a high proportion of them at that point were getting exposed to alcohol, drinking ad lib, not just drinking from a parent's drink, but drinking when they were in high school, or drinking when they were in college or in the military or working someplace. So our number started with 453. We've maintained contact with roughly more than 90% of those original subjects over those 35 years. And as their kids and spouses, et cetera, we now have about 1,600 people as part of this prospective study. As you'll hear, we're interested in characteristics, one especially, which I'll highlight on the next slide. And we're interested in whether that characteristic seen at age 21 was genetically influenced. And because it turns out, yes, but don't tell anyone, it was genetically influenced, but environment was also important. At roughly age 30, we started to be studying the environment as well, and in their kids, we studied the same environmental and gathered blood to look at some of the same genetic characteristics. So we have the 35-year follow-up, and at the 35-year follow-up, because that's where we're going. The end of this talk will be what's going on with those kids and some unique opportunities we have to study those kids, because we've got 2 generations of the same family prospectively. And again, the probans, original subjects. If I slip into jargon, proban means somebody who was chosen to enter the study, 21-year-old student, not alcohol dependent, not alcoholic at the time we studied them, with or without family histories of alcoholism, who were then studied over years to see who developed alcoholism, what I was trying to predict, and who did not. So, looking at the series of steps and kind of boiling them down, things that were there in that prior slide, the San Diego prospective study with the population I talked about, and the characteristic I talked about that turned out to be pay dirt for us, was a low level of response to alcohol. It is when I was in high school in Milwaukee And as a senior in high school, and I'd be at a party, and I'd sit down, and the guy or woman next to me is drinking 6, 8 beers, and they're looking just fine. And I'm having 1 beer, maybe 2, and I'm worried that I'm slurring my speech. And so I have a high sensitivity to alcohol, still drink, but I still have a high sensitivity to alcohol, have to drink slowly and usually with food. And some people have this incredibly low sensitivity where, for example, they could be having 3, 4 drinks in an experiment, and if somebody walked in and looked at our data of what the effect was, they'd say, this guy had placebo, or this woman had placebo, that they're just not showing at moderately low doses of alcohol any major effect. Of course, they have 10 beers and they're drunk, but it is at the lower levels that they're not showing very much effect. And so that's what we'll be talking about. The first follow-up for this study was done about 10 years. It took us 10 years to gather our sample and the testing we wanted to do. And what we found in that first follow-up, you start with people who are drinkers but not alcoholic. Half family history positive, half negative, half family history negative. At the first study, you find out at baseline that those with family histories of alcoholism, and the same is true in Native Americans, and the same is the opposite if you're studying Jews, that we found that a low response, as opposed to my high response, that a low response was characteristic of those with a family history of alcoholism. Could be seen in anybody. It was more characteristic in those who had alcoholic family histories. That's when they're 21. But when they're 30, the low response at age 20 predicted alcohol problems, maximum drinks, and alcohol use disorders by age 30. There were other things that predicted as well, but it was pretty much as good as anything else as a predictor. And it operated despite family history. If you were family history negative, you had no alcoholism in your family, and some of them showed a low response to alcohol, just not many of them. Those with the low response, not showing much effect, and I'll show you what I mean by effect in a minute, you still had a higher risk for alcoholism. It was not the family history per se that was predicting, although that by itself carried some weight, of course, but the low response was predicting, no matter which family history you came from. And since then, we've been gathering data on the course of alcoholism, late development of alcoholism, as we do look at people every 5 years, the environment, and I'll show you what those are that might be important, and oh, thank goodness, we get those kids. Those kids are almost 30 now, which is the age of the first follow-up of those original subjects. So, by background, I knew that alcoholism was genetically influenced when I started the work because children of alcoholics raised by non-alcoholics still have a high risk for alcoholism. Children of non-alcoholics raised by alcoholics, it doesn't seem to affect their risk at all. I'm not saying being raised by an alcoholic doesn't have its problems. Of course it does. I'm just saying that that is not the major risk factor, being raised or not raised by an alcoholic, on whether you'll develop alcoholism. And that it turns out that for alcoholism itself, and it's going to be pretty true of my characteristic that relates to alcoholism, for alcoholism itself, we have a problem as we went from one computer to another. The genes explain about 60% of the pie for alcoholism. And the environment explains about 40%, means they're both pretty darn important. Now, the genes are not going to be simple. This is not alcoholism or this low response, which is one of the characteristics that's carrying the genes. Alcoholism or this low response, they're not affected by one gene. They're not dominant or recessive, as many of us learned in high school or at university. They're much more complicated than that. They're with many different genes, and those genes interacting with environment. So spotting the genes is really tough here. Looking at the genes without the environment for alcoholism per se, or for the characteristics related to alcoholism, is not pay dirt. You got to really consider both of those. So what are some of the predisposing factors toward alcoholism? Let me give you a parallel first. Let's say I was doing this study on heart attacks, myocardial infarctions. It would turn out from the data that heart attacks are about 40% genetic. give or take a little bit here or there, and the rest is environment. And that, what are the genes? Well, there's probably no genes for a heart attack, but there are genes for high blood pressure that increases your risk for heart attacks. And then there are genes for high cholesterol, bad cholesterol, that increases your risk for heart attacks. And there are genes that help protect you because they're high good cholesterol. So you can have a characteristic that isn't dominant or recessive, it's complicated, with many different genes operating through many different characteristics. Well, it's the same thing in alcoholism. And among the characteristics are two that I've been studying over the years. One of them is impulsivity. And you notice some of these are really big because that's what we'll be talking about today. Some of these are also in very light color, but are not quite as big because it's not our focus today. But indeed, if you make decisions without thinking of the consequences, everybody does that now and then. But if that's a regular characteristic for you, oh my Lord, you are at high risk for drug use, alcohol use, gambling. Some will also get involved with criminality. That is a very important risk factor. It's not the one I'm studying per se. It's a very important risk factor, though, and it is not unique to alcohol as a risk factor for alcoholism. But how sensitive you are to alcohol, which is where we're about to go, how sensitive you are to alcohol is indeed just alcohol as the predictor, not anything else, as I said, as I talked briefly before about my follow-ups. So why should a low sensitivity per drink, not feeling much at that first drink and second drink and third drink, why should that increase your risk for alcoholism? Well, here's the theory, and the theory is going to be at least partially right. All right, this is going to shock you. Young people drink for the effects. I mean, that is, we discovered that, but we haven't really published that yet. That if they need more alcohol for the effects, guess what? They drink more. And when they drink more in an evening, they're spending a lot of their time drinking, and they don't hang out with me. They hang out with people who spend a lot of their time drinking. And the heavy drinking itself affects what peers will accept them and who they want, what they expect to happen and how they expect to get to that happiness during a drinking evening, which drink a lot and drink it fast. And that causes stress, and people end up using alcohol to cope with stress. So when we began our studies with these original probands, here's what we did. We took green line people and white line people. Okay, who are they? The green line are family history positive for alcoholism. The white line are people who are drinking about the same amount, or unusual drinks anyway, and same frequency at the time you test them, but they have no family history. You give them alcohol, get their blood alcohol level up to about the level of 3 drinks, which is about 0.06. If the cops stop you while you were driving, you'd be at 0.06%. That about 0.06. And one of the things we discovered about in these earlier probands, that their blood alcohol levels over time were the same, because we'd matched them on drinking. And so drinking by itself will affect how fast you metabolize alcohol, but we're controlling that because our groups are fairly equal, not necessarily on maximum drinks, but certainly on frequency of drinking and usual drinks. Despite the fact that family history positives, high risk, family history negatives, low risk, had the same blood alcohol levels that were very different on effects. Not everybody, of course, but as groups, if we looked over time, that they were given the alcohol, then 60 minutes later, they've peaked on their effect a little earlier than 60 minutes, although their blood alcohol level peaked at 60, and then we follow them to 180 minutes. That as we do that, if we use subjective feelings of intoxication, how high do you feel? It's a more sophisticated series of 13 lines. Then there's a group that's showing a lower response. If we used observer ratings, there's a group with lower response. If we use brain waves, lower response. If we use fMRI, different kinds of responses, these brain wave related. Not everybody has the same markers that are low, but you got a group here, and they are primarily, but not limited to, family history positives who are showing this low response to alcohol. Well, that's all well and good, and indeed, that it was indeed an important part of what we did, but there's a problem with this. This takes all morning. It's expensive, and you can only do it if you live in the city that my lab is in. Of course, other people can do the same kind of thing, but it's going to still be very expensive. The genetic analyses and many of the environmental analyses really require big samples. How are you going to get big samples? Well, now we got a measure. We know the measure from our follow-ups predicts alcohol problems. How might we get a person's level of response to alcohol? There are two measures I'm going to be interested in. But I'd like you all to take notes on this and think about the first. I'm going to ask each of you to stand up. No, I'm not going to. Good. So think back to the first 5 or 10 times you had alcohol on your own, not just a sip of somebody's drinks. Define a drink as a 12-ounce beer, a single shot of whiskey, a 4-ounce wine, which is what they'll generally pour in a restaurant. There are about 5 of those in a bottle of wine. How many of those standard drinks, they all have 10 grams of ethanol. So how many of those standard drinks did it take before you first felt any effect? For mine, a half. That doesn't mean I was drunk. It just means I knew I wasn't drinking water. And then how many drinks did it, by the way, for some people it's 4, to first effect. And how many until you started to feel a bit dizzy or worried you were slurring your speech? For mine, well, it depends for everybody. But generally, if I was sitting and you and I were talking and I had 3 beers by the 3rd beer, I would be worried I was slurring my speech. To stumble, where you're unsteady on your feet, and to fall, I never took enough alcohol to fall asleep, so I don't answer that one. And then if you take my, let's say, 1 drink for this, let's say 3 drinks for that, and it's actually 3 drinks for this as well, that's 3, 6, 7. 3 effects, you divide 7 by 3, and it's 2 point something. And that's your average for the first 5 times. Most people, their average is somewhere 3, 3.5. For many of the people we study who are low responders to alcohol, because now we can do this in thousands of people. Low response to alcohol, many of them it's 5, 6, 4.5 for this first time, first 5 to 10 times drinking. Then these others I don't need you to pay too much attention to, just so you know that I can also look at other time frames in your life where the development of tolerance might have been an issue. And so now we're combining what is probably initial sensitivity with overall sensitivity plus your body changing to resist the effect of alcohol even more. And we call that second one the SRE, because that's the name of this measure. The SRE, self-reported the effects of alcohol. We call it the SRE-T, or SRE total. I'm mentioning that because it's really going to be important, because we're going to compare SRE-5, SRE-T in their kids, from parents to kids, and the impact of it. Now, I was lucky enough to have access to data from a cohort of 8,000 kids in Bristol, England. Following those kids every 2 years, I was just a minor part of that study. And, well, when the kids start hitting age 16, 14, 18, they're drinkers. And so I'm able to, at age 12, take the 8% of the kids who already had some experience with alcohol on their own, not very much, 3, 4 times in their life they drank, average times that they drank was 3, and look at, is there any correlation between how much early in their career they drank per occasion and how sensitive they were per drink? Of course there was, and the correlation is very high. If you have, the correlation is 0.5, thereabouts. That indeed, especially for quantity of drinking, not so much for frequency, not so much for problems, because the low response is predicting drinking a lot per occasion. You can spot it very early. And then we have oodles of studies that have done follow-ups. Does every study show a low response predicts outcome? No, but most of them do. And some in Denmark, some in Australia, some in the United States, just saying, I'm pretty sure low response is predicting outcome. In fact, I'm very sure. So the next thing we said we were going to do over the years is I'll collaborate with people who know how to search for genes. We have a paper now which I'm almost sure is impressed. They asked for some changes. I was a junior person on this paper. The real geneticists were big people on the paper. And that paper to the right here is showing a number of genes that have less than a 1 in a million chance of not being involved with the level of response to alcohol. One of these genes deals with a neurotransmitter, a brain chemical called glycine. Most of them deal with proteins, protein metabolism. I would have never guessed that those genes in white to the right, I would have never guessed any of these genes. Maybe the glycine one, which is this one. But I'd have never guessed any of the genes, and it's done in a non-hypothesis related, something called a genome-wide association study. Another gene variation I'm almost sure of, as related to the low response, is one that involves another brain chemical, serotonin. And the serotonin transporter, which moves serotonin in and out of cells. And I'm fairly sure, although it wasn't part of this study, that it will be important. And I know for sure these guys, which are alcohol metabolizing genes, will have an impact. And those guys got to be controlled. In any of the studies I do, I got to say controlling for the potential impact of these metabolism genes. But probably, if you took all these genes together and you put them together into what's called a polygenic score, maybe I explain 2.5% of the variance. There's too many genes involved. So I say, let me keep searching for genes. They're going to tell us a little bit about what's going on, genes in both the parents as well as the kids' variation, going on regarding level of response, help us to understand the neurochemistry a bit better, but I don't think that's where the money is. I think that it's the environmental measures. So if I have a group of people, let's say about age 20, 18 or 20, and I only take the people who have had some experience with alcohol, which is almost everybody at age 18. And if I take that group and the question is, and if I knew enough about their genes, can I change their genes if they're at high risk for a low response to alcohol? The answer is no. But if I know what's going on in the environment through which the genes are operating, in attitudinal things and environmental things, I might be able to change those. And here are a bunch of things that we did basically in all of our subjects from the San Diego Prospective study and in all of their kids once they began to be drinkers, and in another study I'm about to tell you about. We're interested in drinking history. We're interested in levels of impulsivity. We happen to use something called the Karolinska Institute measure of impulsivity. We're interested in what they think alcohol is going to do to them. Jargon is expectancies, and we use something called the Alcohol Expectancy Questionnaire. We're interested in whether they use alcohol to cope with stress. We're interested in their perceptions of peer drinking. And then, of course, the SRE. So a whole bunch of things. And then how do those things, this is from the San Diego Prospective study as we're working over the years, how do they fit together? Well, LR is abbreviation for level of response. And that it is related to family history. So if you have a family history of alcoholism, you're more likely, but not definite, going to have a low response to alcohol. And as we look at this, the level of response to alcohol by itself is helping to predict heavy drinking and alcohol problems. But the level of response to alcohol is also related to heavy peer drinking through the mechanisms I talked about earlier. It's also related to expectations that alcohol is going to make me fun at parties, join people who are having fun at parties, more positive expectancies of what a drinking, especially a heavy drinking experience was like. And while level of response is related to coping, using alcohol to cope with stress, it's really not much related to stress. Everybody's got stress, and it turns out in these families, which were generally blue-collar, white-collar, pretty functional families, they weren't, the level of response was not much related to level of stress, but how you coped with it. So can I take a group of people with a low response to alcohol who are drinkers but don't have problems, all this coming from the San Diego Prospective study. Can I teach them the dangers of heavy peer drinking and associating with heavy peer drinking? Because it's specifically related to whether they're going to develop problems or not. Can I teach them what are appropriate alcohol expectancies? And can I teach them, for heaven's sakes, don't ever use alcohol to cope with stress, and here are some other coping mechanisms. And that then takes us to the San Diego Prospective study. So we're going to put the San Diego Prevention Study. We're going to put the Prospective study aside, and we're going to come back to it. But a sidelight that occurred is, I got all this information. Let's see if we can put together a cheapo prevention project that we can use with a new population. And that new population is 500 entering freshmen at UCSD. We surveyed several thousand, picked people who, by this self-report measure, looked like they were low response. They needed a lot of alcohol for effect. For every one of them, we picked somebody who looked like they were moderately to pretty sensitive to alcohol, matched them on usual drinking frequency, usual drinking quantity, use of marijuana, smoking, and of course, they're matched on education and educational institution, because they're all coming to UCSD. And among those individuals, for the questionnaire we sent out, we worked hard and we had roughly a 70% response rate, matched the low and high responders, and we put together 3 prevention groups, or 3 groups. Group 1, over the next 55 weeks, approximately every 3 months, a little more frequently, we're going to have you, we'll pay you $25, and we're going to have you fill out a 20-minute questionnaire about your drinking, drug use, and problems. But we're not going to do anything to you. That's the control group. All right? And the control group, half of them are low responders, half of them are controls. Half of them are high responders, I'm sorry. Then we've got a group where 4 videos, me talking, boring, but it is me talking and explaining about the importance of peer drinking, etc. And in this group here, called the LR-based group, every example of things that you can do wrong regarding something that will magnify your drinking was level of response to alcohol. So it was other theories, other roads were talked about, but level of response was really emphasized. And then we had a state-of-the-art group, also watched videos. I made 8 videos, 4, based on the real strong emphasis on level of response, but teaching the same things that the other group got, the state-of-the-art group, except there was no exaggerated emphasis on level of response, and we got the controls. And we followed them 55 weeks. And as Martin said, he didn't say it this way, but he could have said, I'm a pretty obsessive guy. So we got a very high proportion of people who they did what they were assigned to do, and they filled out the questionnaires. And with college students, that is worse than herding cats. It is really something. But it's not brain power, it's just you do the work. And the team that I worked with did a lovely job doing the follow-ups. And let's see what happened over the 55 weeks. So you got the bottom, it's 55 weeks, and then you've got, I can do it with alcohol blackouts, I can do it with other alcohol problems. I'm doing it with max drinks, because maximum drinks is what level of response has its major effect on. Those poor people are the controls. And what happened over the 55 weeks is the very fact that we talked with them, or had them fill out some forms, was associated with this group, which are low responders over 55 weeks that were in the control group. And there was a small diminution, but boy, did they increase. This is known as a mistake. The mistake was that we didn't put in our planning when the university had a celebration called the Sun God Festival, which has a lot of drinking associated with it. But at least it happened to all of the groups. But no matter when you did it, time of heavy drinking on campus or the rest of the year, those poor low responders, a lot of them drinks needed for effects, they didn't do very well. Then this group is the state-of-the-art group. we taught them a whole bunch of things about drinking, but we did never emphasize low response, but it did some work. These are low responders. And then this group are the low responders with the low response-based group. Indeed, replicated this finding. Another group that wasn't quite as obsessive as I am replicated the study at Virginia Commonwealth University. So we're having an impact. Hey, your tax money, which went for the San Diego Prospective study, has developed something that is having an impact. The high responders didn't matter. They never got into, of course individuals did, but those people more sensitive to alcohol really had a much lower rate of, much lower maximum drinks over time and much lower rate of alcohol-related blackouts. Okay, so now we go to the fun part. These illustrations that are artwork, they're all my daughter's. So it is, she's an artist, lives in London, and I love her work, and so I get to use it when I talk. All right, so now, where we are, and this is going to be the final thing that we're going to talk about today, is, boy, we have a unique opportunity. We have, in one set of families, that means we're controlling pretty much for race, we're controlling pretty much across generation for education, because that's the way it turned out, as it should have. We're going to be analyzing data regarding sex, but we are also using the same instruments, such as the alcohol expectancy questionnaire, at about the same age, roughly age 30. That's the age that we picked for the analyses that I'm going to be showing you. And we're going to compare across generation, and these are just those are the same instruments. And you got the same obsessive PI, principal investigator, with pretty much different research assistants, but the statistician has also been with me for 35 years. And so you've got pretty much the same crew that's really handling the data. So the first thing that we took a look at is proband, the fathers, age 30. A few of them had died, very few of them dropped out. We had complete data at age 30 to be using of 439 of 453. And that there were, and it's now larger, by the way, but at least the study that we just recently submitted, there are 216 drinking offspring who were interviewed as this part of the study. And if you take a look, just who are I going to be telling you about across these two generations, the average age is roughly the same, a little bit older, not much for the probands. Education, almost the same. We'll also enter education in any of the analyses we do, but it's not likely to do very much. The original probands or subjects were more likely to have a family history of alcoholism. Why? Because half of them were picked to have a family history of alcoholism. And then when you do a family history study, you generally miss some people who are really family history positive. And so it goes up a little bit with time. But if we were to take a look at that, we'd have said, gee, the probands, original subjects, they were more likely to have a family history of alcoholism. For the SRE 5 or T, I'm just going to give you the 5 here to keep this slide simple. The higher the number, the greater the alcoholism risk. And the probands had a little higher. A number of drinks needed for effect, which means a lower effect per drink. The probands had a little higher rate of maximum drinks, and the two groups were pretty much equal on their experience with non-cannabis. I'm abbreviating cannabis as CB drugs. So if you look at this, you'd say, well, what's going to happen is, of course, if you're looking at number of alcohol problems or people who by age 30-ish meet criteria for an alcohol use disorder, the probands are going to be a bit larger than the offspring, but they didn't. That the proband group, some of whom were alcoholic age 30, some of whom were not, had of 11 DSM, that's Diagnostic Statistical Manual of the American Psychiatric Association, diagnostic criteria set, the probands had less, despite all the rest of this, than did the offspring. And of the proportion of offspring who had an alcohol use disorder, pretty much just meaning multiple of those characteristics in the DSM, it was almost twice as high. Where the heck was that coming from? That was a shocker. I expected that what would happen is, a proportion of people with a low response might change a bit, but that society changes, and it's going to turn out, I projected, I'm wrong at least half the time in this stuff, that I projected that the effect of the low response would be dying out across generations. Well, it didn't happen here. And we got some things here that are pretty odd, that despite heavier maximum drinks at that approximately the same age in the probands, despite a higher family history of alcoholism in the probands, the kids had a higher rate of alcohol problems. What the heck is happening here? So one of the first things that we did, we wanted to look again, proband, again, the original subject, and kids, we want to look at how their SRE scores compared across generations. And it turned out proband to sons are correlating 0.2-ish, which is about where we usually see within families. It's very hard to do probands to daughters because we're dealing with absolute drinks per occasion, and women get a higher blood alcohol level than males. It's a whole different story, but it would have been a wonderful surprise to us if probands would have correlated with their SRE, both 5 and T, the total score, would have correlated with daughters, but they didn't very well. Mothers did correlate very well with daughters, but not so well with sons. But that is quite consistent with there's a genetically related characteristic here. It's just using the SRE. You're going to have to do some controls of controlling for what the usual blood alcohol level could be. But brother pairs correlated almost 0.55. I mean, that's amazing. And then 0.35 for SREG, and sister pairs. Brother-sister pairs, not so good. But what we've shown here is within the same generations, consistent with some other data we had, the SRE is behaving genetically, as if it is indeed a genetically related characteristic. But how about across generations, what's happening with how well the SRE is correlating with how much you drink or how many problems you're having? So here's what we did. The SRE in fathers and mothers correlated with maximum quantity pretty well, but even higher in the kids. You look especially proband to son, again, now it's more than a doubling of how well did the SRE score correlate with how much somebody drank per occasion? In the kids, it was much stronger than it was in the fathers. And in the daughters, it was still higher, was statistically significant, but not so much higher. So why are the kids who have similar SREs, which we've just shown you, as their parents, having a higher rate of alcohol problems? Part of it is the SRE is behaving more strongly related to alcohol problems in the kids than it did in the parent. SREs weren't all that different, but the correlation with maximum drinks and alcohol problems were different with a higher impact, especially for boys in the next generation. Well, why the heck is that happening? So what we did is next, we basically tried to follow up, this is just a repeat of a slide, of where this higher rate of alcohol problems is coming from in the offspring. And the first thing we did is we say, okay, let us take, the ends change here because there's a variety of reasons of some of these analyses required that you have same offspring and parent. Here it's not necessarily required. And so what we say is, okay, we got all these probands, we got these kids, just what did they differ in? And they differed in, the kids were more likely to be smokers. The kids had a little higher, this is significant because of the very small standard deviation in this test, that had a little bit higher impulsivity. And the kids had one of the alcohol expectancy scores, which relates to that it will make you have more fun, was higher in the kids. But the cannabis use doesn't, use of drugs other than cannabis, it's higher, but not in a way that's likely to explain why kids are having higher rates of alcohol problems. And the peer drinks are almost identical. If anything, it's a little bit higher in the kids. Well, that's one way to do it. You can say, all right, I got 2 generations. SREs are pretty similar. One generation, unexpectedly, is showing a higher rate of alcohol problems. Let's just compare the 2 generations. And we come up with a hint that maybe impulsivity is important, maybe smoking is important, which is, actually, I take it wrong. I'm sorry. Maybe smoking is important. And then the social behavioral attitudes towards the drinking. Maybe it's contributing. So the next thing we did is we say, okay, let's do what's called a regression analysis. Let's look within probands, what about them predicted their number of alcohol problems, and within kids, what predicted. And what we end up with here is family history is pretty similar, but SRE5 is predicted. We knew that from some of the other analyses that we were doing. That wasn't a surprise. And indeed, it shows up here that SRE5 is much stronger related to alcohol, 4 times in this particular analysis. Within kids, it's a better predictor than of alcohol problems than within probands. So is surprising to us, because it didn't show up all that much earlier, was the use of non-cannabis drugs. Impulsivity only predicted in the kids, not in the parents in these analyses. And there's your social behavioral score again. Now, the last thing I'm going to take you with, again, we're looking at an unexpected higher rate of alcohol problems in kids compared to the original probands. And we're coming up with some hints here that it's probably somewhat related to SRE being more powerful in the kids, which is probably because of this social behavior expectancy, because expectancy itself is one of those mediators of how level of response finds its way. That this small increase in impulsivity in the kids is probably contributing, and it is potential that use of drugs other than cannabis is predicting, even though it was more common in the parents. When it's there in the kids, it is more likely to be predicting the outcome. Last thing. We did something with a statistic called R. And in the R statistic, we did 1,000 random matches of each proband with an offspring. And then we would take those pairs, look at what predicts, look at this again, but now within those matched pairs. Then throw it away and go to another proband, different proband, and randomly match him with somebody else. And then do it 1,000 times. And then we would look at the pattern across these regression analyses. And what that pattern showed us is the following. That what it's demonstrating to us is done 1,000 times the way it was, excuse me, I'm so sorry, that done 1,000 times, that SRE, now this is all offspring, male offspring, female offspring, because it's the outcome score is the difference between the kids and the fathers. So the fathers they're already considered in here because it's the difference score between the two using this crazy statistic. That it turns out that we can explain about 35% of what's happening here, of the difference. That's the R square, it's called, which is roughly a measure of how much you're explaining. And SRE5 is the most powerful of all these predictors. Social behavioral expectancy is the next most powerful. It turns out that non-use of CB is indeed powerful, and impulsivity still maintains its power. Family history of alcohol use disorder we never picked up before in any of the other analyses. I don't know whether it's really doing it or not. And that's the same thing for pure maximum drinks, which never turned out in any of the additional analyses to be very important. But to end what we're talking about today, my conclusions are 35-year studies across generations are difficult to do, but I've had a ball doing them. And that While the people at the National Alcohol Institute said, after our probans reached age 45, they said, what are you going to get from this study? So why don't we give the money to somebody else? And I've been doing the remainder of the study through private money, through donations. I'm not going to ask. But I've been doing it through donations. And what we've discovered regarding the kids is just fascinating. And if we're right, if somebody can replicate this, to find out that it is changes in attitudes. There are a lot of things that changed across the generations, but it's changes in attitudes about social behavioral impacts of drinking to the positive side that we really should be working with kids. If there's any truth here, across the country, we've got to do something about those social behavior belief scores because they're impacting on other risk factors as they are going on to severe alcohol problems and to heavy drinking. So it's valuable, it's challenging, and it's a unique opportunity. When we started to do these analyses about why the difference was there, I'm just saying to myself, my God, I can't believe we have the opportunity to, right or wrong, but at least test this to try to figure out what's going on. So the number one is these are challenging. We can uniquely, not telling you more truthfully than others or with greater level of truth, but we have a unique advantage at how we can look at some of these characteristics that are associated with increased rates of alcohol problems in younger generations. It's happening here, it's happening in San Diego, and it certainly was picked up in the San Diego Prospective study. And in the meantime, as I said to some of the students and fellows and postdocs that I've been meeting with here, man, if you're not having fun when you're doing this, find something else to do, because one of the things that keeps me going in research is how much fun it is. And the wonderful people I meet, like Martin Paulus, the places I get to visit, such as this, and the pleasure of presenting this information to an audience such as you. So thank you. Thank you. Thanks. So now we get questions. This is my favorite part. Anyone have any questions? That's a lot of stuff I gave you, because this was like a Whitman sampler kind of overview of what the work was. Yes, Martin.
So you talked about the changing attitude and expectations. Would you be able to describe what is? Yeah. Okay.
I like standing there.
So you talked about the changing attitudes and expectations.
Yes.
Would you describe what the changes are? What are the different expectations that the youth has now that the probans did not have in the beginning?
Super. And it's not that they did, and it's a wonderful question, and it's not that the probans didn't have it, they just didn't have it to the same extent. And by the way, I glossed over male-female differences. There are some, but the basic findings are very similar for males and females. But there were some, and the paper takes care of that, discussing them. There are, I think, 6 questions, I don't remember exactly, on the alcohol expectancy questionnaire regarding how my social behavior is affected by alcohol. And they included things like, if I'm not drinking, I can't join in the fun other people are having. And things like the best way, a related question, the best way to have fun at a party is to drink a lot. Again, those aren't the exact words. But heavy drinking appears, and this, by the way, is good for the data that they're calling binge drinking, heavy episodic drinking. Heavy drinking has always been a problem. It's been a problem of youth. And it is apparently worse now, and it's worse enough now that if you consider age, you consider education, you consider impulsivity, you consider level of response to alcohol, and all of the variance taken up by those when you do these regression analyses, that one additional characteristic came up in all of our 3 ways of looking. And where is that coming from? I can make some guesses, but I don't have any data.
Well, so I want to follow up on this a little bit because I've also talked to some other people who are doing studies at UCSD, and what they have observed, and this is very speculative at this point, is that we seem to have an increase in social anxiety, and that the levels of social anxiety.
Let me see, who are you talking about? Because we have it at UCSD, the world-class guy on social anxiety disorder.
But what I'm wondering is, with these alcohol expectancy, I'm wondering that in a sense what your experience is people drink to relieve their social anxiety. Not that as a treatment,
Yes.
but they drink so they don't feel socially anxious so they can engage in...
Right on. I'm going to admit something that I hope there are no reporters here. I drink at parties, at least in part because of who likes walking into a party? There's a lot of noise going on and a lot of, but I stop. And it's probably, it can't just be the social anxiety. And your anxiety gets worse, at least the next morning, if you're drinking too much. So we could say social anxiety is contributing to it, discomfort in large groups, perhaps related to spending too much time on the telephone and too much time on the internet. But whatever it happens to be is almost certain, would not surprise me to be real. And it is, I think, a very common reason people drink alcohol at parties. One of the things is I have a natural stop, which is I don't feel good. Anyway, when it comes to something that embarrasses me, I'm close to a one-time learner, and I don't want to do that again. So I have natural stops. We may be starting drinking for the same reason. I have a natural stop, and at least theoretically, the people with low response don't have a stop until they get up to doses of alcohol, which is really hard to control anymore because you've got a large volume still coming through. But I like the thought, yes. Yes, what?
The price of alcohol, how does that compare? And do you think increasing the price of alcohol would help at all?
Yes. We come to one potential conflict, because now we're talking about pricing, and that I run an educational program that is underwritten for the University of California by Anheuser-Busch. It's an education program. It's nothing to do with anything else. And I am sure Anheuser-Busch, if they had any influence over me, and they don't because they don't touch this stuff, would want me to say taxation doesn't have much to do with it, and I would say, pooey, that's wrong. Taxation has a huge amount to do with who drinks and how much they drink. And the good thing about my work is taxation has changed over the years. It's gone down in the U.S. Dramatically per drink. As inflation gone up, the taxation has stayed basically the same. And that may have something to do with drinking overall, but has nothing to do with my prospective study, because everybody, low response, high response, kids, whatever, and among the kids, they're all studied under the same kind of time frame. But yes, well, I don't think it's directly related to my work. I think taxation is way too low, and that it has a lot to do with who drinks. Trouble is, my understanding of that literature, and I'm not an expert on that literature, is it seems to have its greatest effect on the people who drink the least. The people who are really boozing it up, price is almost inelastic, is almost elastic. It doesn't seem to have much effect. Yes?
In a word, availability theory. The more available it is, the more it is.
Oh, and I fully agree with that. So again, taxation has something to do with availability. Has a lot to do with availability. In my life, it wouldn't have an effect. I mean, I'm very lucky. I married an heiress. No, I did not. She's not an heiress. But I'm very lucky that it wouldn't impact. But for the most of people in the United States, it will have an impact. But the real heavy drinkers, it looks like they keep doing it despite the cost. Maybe they just go to cheaper alcohol. Yes. And you can speak loudly if you can.
I was just wondering if there are any interesting psychological correlates of levels of responsiveness.
Okay.
Looked at
Okay. Question is about a whole area of research that we certainly have been, that we have, the area of research that we've been interested in, and it is level of response to alcohol. What psychological characteristics is it related to? It is not related to impulsivity. The correlation is about 0.1, so it's not zero, but it's not very high. It is maybe a little more related to sensation seeking, but not very much. Doesn't seem to be related to conscientiousness overall. So if you look at the NEO or you look at many of the personality characteristics, there's not a huge amount there that it seems to be related to. We know it's not related to mood. We know it's not related to risk for psychiatric disorders. It is, and in the models that we use, while I didn't show you, we've looked at in the probands and in the offspring and in the collaborative study on genetics of alcoholism and in LSPEC, this Bristol study, we've looked at the relationship between time 1 impulsivity measure and level of response measure. Various mediators as time 2, as time goes on, and the outcome of heavy drinking. Both contribute to the model, impulsivity and low response, but they have somewhat separate paths on the way to the outcome. So I don't think this is closely related to at least any personality characteristic we've been able to study. We'd be happy either way, but it doesn't seem to be. Other questions? Yes? Is higher. Well, I can't, the question is, I'm going, well, no, I think that's a smart question. And the question is related to, when I showed slides, there were a bunch of characteristics that I thought would be related to heavy drinking that were higher. And I don't remember specifically here, I have it on the slide, but the cannabis, use of cannabis or use of drugs other than cannabis or cannabis use disorders, some of them were higher in the probands. Could that have been protective? I have no data to say that it's protective, I have no data one way or another. And when I focus, and that's the other thing about my obsessiveness, when I focus on what is the question I'm asking, the question right now I'm asking is, why was there that unexpected higher rate of alcohol problems in the kids, in things that I might be able to do that might be able to interpret more directly? And so I paid attention to those things. It could have been higher in the proband, but the real question is, did it contribute to the outcome? And cannabis came nowhere in contributing to the outcome. And if it had a significant protective effect, I might have expected to see it. But I'm focusing on another question. And did not contribute. The question is, there were also things that were much higher, or were higher anyway, significantly higher, in the offspring, but didn't contribute within kids or probands, and did not contribute to this difference across. And so I'll take smoking as the example and say, I haven't proven it's not important. I've just proven within this population, now we're talking Southern California to start with, they're all over the country, but Southern California to start with, where smoking is not a lot of people smoke. And it could be that in another population, it would be important. But within this population, despite that difference of higher smoking in one group than the other, it did not enter any of the things where I'm looking at predictors of the outcome. And especially the granddaddy, which is that last 1,000 bootstrapped, with 1,000 variations or 1,000 reiterations or iterations of it, it didn't enter at all. So if we come to, and again, it's a nice one to close with, because we've come to one of the questions that was coming up when I was talking with other people this morning, which is, nobody does a perfect study. I certainly haven't. and that all of the studies add some interesting information. Whatever that information, potentially, and whatever that, if the study's done moderately well, whatever that information is, could be just luck. You got to see if it's replicated. I probably was doing this work with level of response for at least 10 years before I started to believe what we were seeing was more than just some mistake we made in how we did our analyses or whatever. And that no matter what it is you study, there are hundreds of things that could be important that you didn't put into your study. And so I just take what I get based on what I was able to ask. And here I am moderately convinced, I'll bet at least $100, that that indeed those characteristics, maybe 50, those characteristics that I'm saying are probably contributing to the higher rate of alcohol problems within this population of white collar primarily and blue collar, relatively highly functional families, I'll bet that's real. And I'll bet it's the tip of the iceberg, that there's a whole bunch of other things. So again, I'd like to thank you all for being here, and Martin for inviting me. Thank you. Thanks.